Hello guys, I am Harun and it's my YouTube channel Harun Biozone. Today in this video I will discuss 43 very important MCQ questions from the chapter Molecular Basis of Inheritance. Okay, so let's start the video. The first question is, what is the distance between two base pair in the double helical model of DNA? In the double helix DNA model, the distance between two base pair is 0.34 nanometer. The A option is correct. Let me explain. Here the double helical structure and the distance between two base pair is 0.34 nanometer and the complete turn of DNA is 3.4 nanometer and total number of base pair present in complete turn is 10.5 base pair. Okay, and the radius of the DNA is 2 nanometer. The radius of the DNA is 2 nanometer. And distance between two base pair, suppose this one base pair and this one base pair, this base pair distance is 0.34 nanometer. Clear? So here the right answer is 0.34 nanometer. Next question, deletion or insertion of 3 or its multiple base results in frame shift mutation, point mutation, no effect on reading frame. The frame shift mutation is occur by point mutation or gene mutation by the addition or deletion of a single nitrogen base to the DNA sequence, but here no effect on reading frame. How? Suppose this is 3 nitrogen base pair. Okay, AUG, GUC and CUG. If 3 nitrogen base or its multiple produce one codon, if this codon is changed, suppose AUG and GUG, then CUG, the number of codon do not changes. The number of structure do not changes. Okay, so no effect on reading frame. Let me explain. Here, so frame shift mutation. Frame shift mutation means when the single nitrogen base addition or deletion to the DNA sequence. This is called frame shift mutation or point mutation. Suppose cat eat the rat. Suppose cat eat the rat. Say for example, here 3 nitrogen base, so produce one codon each. If one nitrogen base is added, suppose this nitrogen base is deleted, then what happen? CAE ATT HERAT. Okay, so some completely change by addition or deletion of a single nitrogen base. So all total frame is changes here. So it is called frame shift mutation. But in case of if 3 nitrogen base means one codon or multiple of one codon is completely changed, there is no effect on reading frame. Okay. The next question is, the experiment by Hershey and Chase helped to confirm that DNA was the hereditary material on the basis of the finding that radioactive phases were found in the pellet. Okay, here Hershey and Chase used two radioisotopes S35 and P32. Okay, the S35 sulfur is the component of protein. Protein means the outer structure of virus, bacteriophage is made by capsomer protein, and in capsomer protein the sulfur containing amino acid cysteine and methionine present. So sulfur are attached to the capsid region, okay. And P32 phosphorus is the component of DNA. Phosphate is a component of DNA. So P32 bind with this DNA. The S35 bind with capsomer and P32 bind with DNA. The radioactive phases were found in the pellet. What is the pellet means? Suppose this is the centrifuge tube. Okay, centrifuge tubes, here liquid are present or cell suspension are present or bacteriophage are present. Okay, then it is centrifuge. If this tube is centrifuge, then produce the debris are deposited in the lower side. This is called pellet. And the supernatant, this is called supernatant. The liquid portion is called supernatant. Okay. The radioactive phases were found in the pellet. Radioactive cells were found in the supernatant. Radioactive sulfur was found. Radioactive, not radioactive phase. Here use radioactive sulfur or radioactive phosphorus. The radioactive phosphorus was found in the cell. Means suppose this is a bacteria. When bacteriophage attack, the DNA bacteriophage transfer is genetic material, DNA material into the bacteria. Okay. So when the centrifugation occur, the bacterial cell contain this radioactive phosphorus, not radioactive sulfur, because the coat protein remain outside of the bacterial cell. So here bacterial DNA, the viral DNA, and capsid protein remain outside of the bacteria. So S35 do not express here. Here we express P32. When we centrifuge this bacterial cell contain radioactive phosphorus. So here the right answer is D. And this is the Hershey and Chase experiment. Okay. Next question is, during analysis of the DNA of an organism having 5386 nucleotides, adenine 29%, guanine 17%, cytosine 32% and thymine 17%. According to Kargov's rule, adenine plus guanine equal to cytosine plus thymine, or adenine guanine by cytosine plus thymine is equal to 1. This is the Kargov's rule. Or according to Orson and Crick model, adenine always bind with thymine and guanine always bind with cytosine by 3 hydrogen bond. So here if this is a double helical structure, here if one strand contain adenine, then definitely the opposite strand contain thymine. So when adenine, thymine are equal in number, equal is percentage, that is double stranded DNA. If the adenine concentration or thymine concentration or guanine concentration and cytosine concentration are different, that means this is single stranded DNA. So here adenine 29% and thymine 17% means their adenine, number of adenine and number of thymine are different. The different means it is a single stranded DNA, not double stranded DNA. Next question, if the length of the E. coli DNA is 1.36 millimeter, then how many base pair are present in E. coli? So we know the length of the DNA, equal to distance between base pair, into number of total number of base pair. Total number of base pair. So here present length of the DNA present. Now how many base pair is present in the length of the 1.36 millimeter. Distance between base pair is 0.34 into 10 to the power 9. 10 to the power 34 into 10 to the power minus 9. And number of base pair x. Okay, the x equal to 1.36 by 0.34 into 10 to the power 9. Clear? So here the right option is 4 into 10 to the power 6 base pair. 4 into 10 to the power 6 base pair. If you calculate, find 4 into 10 to the power 6 base pair of DNA are found. Clear? Right option is C. Next question, if the sequence of bases in coding strand of DNA is ATT, CG, ATG, then the sequence of bases in mRNA will be. So suppose during transcription process, this is the transcription bubble. Okay, transcription bubble. The coding strand of DNA contain adenine, thymine, cytosine, guanine, adenine, thymine and guanine. Okay, the opposite strand definitely on thymine, adenine, guanine, cytosine, thymine, adenine and cytosine. So if the coding strand, suppose this is one, this one is coding strand, this one is coding strand. The complementary strand is this one. So if the mRNA is synthesized, then what happen? T opposite of T is A. Opposite of U, G opposite C, G, A, U, C. A, U, C, A, U, C, G, A, U, G, A, U, G, sorry, G. So here the right option is D. Okay. The mRNA sequence is just sequence of mRNA is a duplicate copy of coding strand. Okay. In this way, the information or code that present in DNA are transferred from DNA to mRNA. Okay. Next to protein. Next question is, if the base sequence of one strand of DNA is CAT, TAG, CAT and GAC, what would be the base sequence of its complementary mRNA? Complementary mRNA. Same type of question also. This is the transcription bubble. Okay. And if the DNA sequence what happened? CAT, if the DNA sequence CAT, TAG, CAT and GAC. Okay, this is the sequence of DNA. This is DNA sequence. If the mRNA is produced, then produce G, U, A, U, C and G, U, A, G, U, A and C, U, G. Okay, the G, U, A, U, C, G, U, A, G, U, and C, U, G is the right option. This is the complementary. This is the complementary mRNA. Complementary mRNA. Clear? Next question. The function of nonsense codon. What is the nonsense codon? Nonsense means the codon that do not bear the code information of amino acid, or the code do not code for amino acid. UAA, UAG and UGA. 3 codon. This is called stop codon or nonsense codon. The function of nonsense codon. So what is the function of nonsense codon? Nonsense codon, the function of nonsense codon act as a termination codon, or where the termination process is stopped or protein synthesis process stopped. So to release the polypeptide chain from tRNA, to form an unspecified amino acid, to terminate the message of gene control protein synthesis, to convert sense. So here the right option is to release polypeptide chain from tRNA. When the translation occurs, this is the mRNA. Suppose this is the start codon AUG, suppose GUC, and here the one stop codon is present. Okay, so here the suppose this is the small subunit ribosome, this is a large subunit of ribosome. The polypeptide chain are produced here, but when this stop codon do not bear the code of amino acid, so when the ribosome reaches or the A site reaches to the UAA region, there is no amino acid, so no tRNA is bind. So in this place the polypeptide chain are detached and each of the part of the RNA translation are detached from each other. So translation is stopped. So to release the polypeptide chain from the tRNA, this is the right option. Next question is, the promoter site and the terminator site for transcription are located. Suppose this is mRNA. Okay, this is one this is the one DNA, double stranded DNA. Okay, so this is the suppose this is the transcription initiation site, transcription initiation site. Okay, so here from the transcription initiation site, it is 5' region, this is 3' region. Okay, so transcription occur from this region to this region. Okay, so the opposite of the transcription initiation site or towards the 5' end region is called upstream region. Upstream region, and from transcription initiation site, this region is called downstream region. Okay, this is symbolized by negative and this is symbolized positive. The promoter region are present in such region, -35 base pair region or -10 base pair region. Okay, Tata box or Pibno box etc. are present from transcription site. Okay, this promoter region means where the RNA polymerase bind. Okay, -35 base pair means from the transcription initiation site 30 base pair upstream region the promoter sequence are present. Okay, this is upstream region, so the 5' is upstream region and 3' is downstream region. The promoter sequence present in the upstream region, and terminator means transcription termination site. Suppose transcription is stop here, terminate here. So this is called termination site, transcription initiation site, termination site. The transcription termination site present in the downstream region and transcription promoter site is present in the upstream region. This is the mRNA and this is the promoter sequence where the RNA polymerase are bind, and this is a transcription initiation site from where the transcription start towards side. Okay, so in this region is called upstream region, this region is called downstream region. Clear? This upstream region the promoter is bind, and this promoter then move into the transcription initiation site and from where the transcription starts. So here right option is B. Next question, a messenger acid is 336 nucleotides long. Okay, 336 nucleotide Okay, so on 3 nucleotide produce 1 amino acid. So if divide, then 1 1 2 amino acid are produced. From 336 nucleotides, 112 amino acid are produced, including initiator and terminator codons. Okay, initiator, terminator, the number of amino acid in the protein. Terminator protein means, terminator means UAA, terminator means the nonsense codon, okay, or stop codon, UAA, UAG and UGA. Clear? The terminator codon do not bear any information about amino acid. So here 336 nucleotide, okay, produce 100, produce one amino acid, because one codon, this three, because three nucleotides do not present any amino acid. So 336 minus 3 means 333, only 333 nucleotides produce 111 amino acid, and remaining 3 codon represent one termination or stop codon that do not represent any amino acid. Clear? So here the right option is 1. Next question, total amount of adenine and thymine in DNA is 45%, amount of guanine will be the adenine plus thymine 45%, the remaining 100 minus 45, that is 55%. So 55, within 55% guanine present and also cytosine present. The guanine equally bind to cytosine, so divided by 2, that is 27.5% guanine and 27.5% cytosine are present. The right option is D. The amount of guanine is 27.5. Next question is, match the column and select the correct option from the codes. Frederick Griffith. The Frederick Griffith experiment on mice to prove the DNA is a genetic material. So Frederick Griffith doing the experiment phenomenon of transformation. Okay, Gamo in the Watson and Crick double helical model, Michelson installed semi-conservative mode of replication, and Gamo, a sequence of 3 nucleotide codes for a single amino acid, that is genetic code, genetic code. B. Gamo. So here Frederick Griffith 5, that is phenomenon of transformation, that is DNA as genetic material, DNA as genetic material. DNA genetic material. Then Gamo is genetic code. Gamo represent genetic code, that 3 nucleotide represent 1 amino acid. Watson and Crick double helical structure. Michelson installed semi-conservative mode of replication of DNA. So right option is A. Next question, match column 1 with column 2 and select the correct option from the codes given below. So sigma factor is a initiation of, so this is RNA polymerase. In RNA polymerase there is alpha 1, alpha 2, beta 1 and beta 2. 4 base, 4 units are present, and this is the sigma factor. Okay, this sigma factor attach with RNA polymerase 2 and produce a holoenzyme. This holoenzyme bind the this sigma factor helps to bind this RNA polymerase II to the initiation site during transcription. So, sigma factor transcription, initiation. Capping means if this is mRNA. In case of eukaryotes, when the mRNA transformed from nucleus to the cytoplasm, the 5' end is capping with 7-methyl guanosine cap. Cap means catabolic activator protein, and 3' end is 200 to 250 amino adenine, okay, polyadenylation, okay. This is a modification of mRNA, So, capping is 5' end. Tailing means this is polyadenylation or 3' end, okay. This is tailing 3' end. And coding strand means this coding strand from the region of mRNA where translation occur, that is 5' to coding strand present from 5' to 3'. So, here A is 2, A2, B4, C5 and D1. So, right answer is B. Next question. Go through the following statement and select the correct option. In DNA replication, the primer is a small ribonucleotide polymer. 100 to the RNA primer that used in lagging strand of DNA during DNA replication is 100 to 1000 base pair long. Clear? DNA act as template for synthesis of both DNA and RNA, because DNA produce DNA by replication, okay, and DNA produce mRNA by transcription. So, in both cases the DNA act as a template. So, this option is also correct. RNA attach amino acid at its 3' end. Here attach means this is clover leaf model of tRNA, and this is the site of ACC. This is 5' end, this is 3' hydroxyl site from in where the ester bond is formed, okay, or peptide bond is formed and produce, and this is amino acid binds here. The RNA attach amino acid at its 3' end. Blender experiment. What is blender experiment? Blender experiment means Hershey and Chase experiment. Chase experiment for prove the genetic material, DNA as a genetic material. So, this is the wrong answer. Here Watson and Crick is not right. Here Hershey and Chase. So, only D is the incorrect option. Next question. Read the following statement and select the correct statement. From this given option. The chromatin that is more densely packed, and stained dark are called euchromatin. So, at the time of interphase, the euchromatin are lightly stained, but in case of cell division, the euchromatin are darkly stained. Okay. So, this is the wrong option. Euchromatin is said to be transcriptionally active chromatin. They have been right euchromatin because euchromatin carry most of the coding region or exon. Most of the exon present in euchromatin. Oswald, Avery, MacLeod and McCarthy ought to determine the biochemical nature of transforming principle in Griffith experiment. Definitely they use heat killed S3 strain, heat killed S3 plus R2 strain, okay, and inject mice. Before this they use 3 enzymes, DNase, RNase and protease. When they use DNase, the mice do not die. When use RNase, mice die, and RNase use, mice die. Protease, mice die, but DNase do not die. Means the transforming material is DNA. When use DNase, the DNA is destroyed. So the strain become non-virulent. Okay, the transforming material is DNA. This is the right answer. The unequivocal proof that DNA is the genetic material come from the experiment of Alfred Hershey and Martha Chase. Hershey and Chase used a radioisotope to prove that DNA as genetic material. This is also right. So here the correct option is B, C and D. Next question. In a piece of DNA, adenine 22.4, cytosine is 31.3%, guanine is 30.4% and 5-methyl uracil 32.9%. DNA polymer is 31,005 nucleotides long. Find out the most relevant conclusion for the given data. The most relevant conclusion is here adenine, here guanine 13.4%, cytosine is 31.3%. That means guanine is unequal to cytosine. That breaks the Kirchhoff's rules. So this is a single stranded DNA is a single stranded. Okay. In case of double stranded, number of guanine and cytosine are equal. So here the right answer is DNA is single stranded. Next question. The recombinant frequency between the genes A and C is 5%, B and C is 15%, B and D is 9%, A and B is 20%, C and D is 24% and A and D is 29%. What will be the sequence of these genes on a linear chromosome? So according to chromosomal theory of inheritance by Sutton and Boveri, the genes are present on chromosome linearly, that is A, B, C, D. In this type of question, we first observe the long distance. Okay, so here suppose this is a chromosome, the most distance is A and D. So A and D is 29%. Okay, then 24% distance is between C and D. So C and D is 24%. Okay, then 20% A and B. A and B is suppose 20%. This is B, 20%. Then 15% B and C. This is 15%. B and C is 15%. And B and D. B and D is 9%. Okay, and then A and B. B and C is 5%. A and C is 5%. A and C is least 5%. So here the arrangement is A, C, B and D. ACBD, this is the right answer. So here in this type of question, first observe the maximum distance, okay, sequence from maximum to minimum, okay. Next question is, the length of DNA molecule is 1.1 meters. What will be the approximate number of base pair? So we know the total length of DNA, total length of DNA equal to number of base pair into distance between base pair, So here length of the 1.1 meter, clear, and number of base pair we know that 0.3, number of base pair, distance between base pair is 0.34 into 10 to the power 9, okay, 10 to the power 9. The number of base pair x, so x equal to 6.6 10 to the power 9 base pair is the result. If we calculate B 3.3 10 to the power 9, B is the correct option, okay, 3.3 into 10 to the power 9 base pair, if you calculate it. Next question, 10 E. coli with N15 double stranded DNA are incubated in medium containing N14 nucleotide, okay. 10 N15 double stranded DNA are, first this is N15 red in color, suppose N15 DNA, clear, this is N15. Then it transfer into the culture medium containing N14. N14 means blue in color, okay, N14 blue color. So here in this culture medium, when N15 is cultivated, the in first generation, suppose this is N15, clear, culture in N14. So here the DNA strand is separated and new strand is synthesis from this medium. So here one strand is synthesized. Again this is first generation. Second generation, in case of second generation, the DNA strand is separated in N14 medium and produce new strand of DNA, and also, but here one parental DNA is present, clear, this is second generation. Here 2 DNA, here 4 DNA, okay. So here 60 minutes means generation time of E. coli is 20 minutes, clear. The 60 minutes, 60 minutes means 3 generation, the 2 to the power 3 means 3 is the number of generation, total number of DNA produced 8, clear. The 10 E. coli means 10 into 8, 80, total number of cell 80 are produced. In 18 cell, 16 cells are N14 medium containing and 20 cells are N15 medium containing cells are produced, okay. The right answer is 60 cell. The 16 cell totally free from such type of cell, 16 cell are produced, and such type of cell 20 cell are produced. Next question, the codon AUG codes for methionine and phenylalanine. First AUG code for methionine, this is called start codon, start codon. This code for methionine. But phenylalanine means UA, UU, UUU and UUG, UC. This is the code for methionine. This statement is wrong. AAA and AAG both codes for the amino acid lysine. This is right. So here statement 1 is incorrect, but statement 2 is true. Next question. If the distance between two consecutive base pair is 0.34 nanometer and total number of base pair of DNA double helix in typical mammalian cell is 6.79 base pair, then the length. So here length of the DNA, length of the DNA is equal to distance between base pair and total number of base pair So here distance 0.34 into 6.6 into 10 to the power 9. This if you calculate it, the option is B, 2.2 meters. Next question. Choose the correct pair. Polymerase. Polymerase means synthesis DNA. Synthesis DNA. So break the DNA fragment? No, break the DNA fragment by endonuclease. So this is wrong answer. Nuclease. Nuclease breaks the DNA. Okay. Nuclease destroy or break nucleic acid. Clear? Now separate the two strands of DNA called denaturation. Clear? Denaturation depends on pH, temperature, etc. Determine the denaturation of the DNA. So this is wrong answer. Exonuclease. Exonuclease means exo or remove nucleotide from 5' or 3' end of the DNA. Okay. Remove nucleotide from 5' or 3' end of DNA. Make cut at a specific position of the DNA. This is called restriction endonuclease. Endonuclease cut a specific position of DNA. But ligase join two DNA molecule by phosphodiester bond. Suppose this is one nucleotide. Okay, this is O. This is joined by OCH2. Clear? So there this 3' and this 5' end are jointed by this phosphate. Phosphate come from ATP phosphorus, ADP plus phosphate. This phosphate and this reaction is caused by the ligase enzyme. Clear? So here the right option is ligase. Next question. If adenine makes 13% of the DNA molecules, what will be the percentage of thymine, guanine, cytosine? So total percentage, 100%. The adenine is 30%. According to Kargoff's rule, adenine plus guanine, cytosine plus thymine, okay, where adenine equals thymine and guanine equal cytosine. The adenine 30% plus guanine, cytosine plus 13%. Okay, so we can write here plus guanine plus 13%. Clear? The 2 guanine, so here remaining 30% will be adenine, 13 with thymine 30%. The remaining 40%, so within 40%, so guanine is 20% and cytosine is 20%. So this is B is the correct answer. Next question. Escherichia coli fully labeled with N15 medium is allowed to grow in 14 medium. The twist strand of DNA molecule of the first generation bacteria. So suppose N15 medium, this is N15 medium, red is N15 medium. If you transfer to N14 medium, so what happened? The twist strand separated during first generation, twist strand separated and produce new strand from N14 medium. So this is okay. So in first generation produce half of half is N15 and half is N14. So N14 and N15, N14 and N15. So the right option is B, different density but resemble parents. DNA, different density. Density different means N14 and N15. Different density means if the DNA strand is centrifuged, then two in the supernatant region, one strand is N15. Okay, one strand is N15 and another strand is N14. Okay, N14, N15. So different density but resemble parent DNA, like. The B is the correct option. Next, select the twist statement out of the four given below the lack operon. So what is lack? First, what is lack operon? In case of lack operon, this is the DNA, okay, where one is repressor region, promoter region, operator region, and this is a structural protein ZYA. Glucose or galactose may bind with the repressor, and so here produce repressor protein. This repressor protein is repressor protein. Okay, this repressor protein bind with the operator region, so the RNA polymerase do not move, so no transcription occur. The glucose or galactose may bind, not glucose or galactose. This is lactose. In the absence of lactose, the repressor binds. In the absence, because lack operon is inducible operon. In the presence of lactose, the operon is run and through transcription translation, the product is produced. So when the lactose is absent, the repressor bind to the operator region, so the RNA polymerase do not move from promoter region to this structural gene. So no transcription occur. But the Z gene code for permease. This Z gene code for beta galactosidase. And Y for transacetylase. And this A for permease. Okay, the beta galactosidase permease is A. This is the wrong answer. This was elucidated by this operant model was first observed by Jacob and Monod. This is right. So 2 and 4 is the right answer. Next question is, lac operand is seen in E. coli, right? Lac operand seen in E. coli. E. coli lacks a definite nucleus. Definitely, E. coli is a prokaryotic cell, so no nucleus is present. So both assertions are reason are true, but reason is not the correct explanation of assertion. B is the correct answer. Next question, the basis of DNA fingerprinting is. So what is the DNA fingerprint? DNA fingerprinting means it is a forensic technique by which the crime analysis are done here. Okay. So in this DNA fingerprinting technique, they use RFLP. This is they use RFLP, restriction fragment length polymorphism. RFLP is a technique. RFLP is a technique. Restriction means the DNA first they collect the DNA by then cut the DNA with R for restriction endonuclease. Cut the DNA with restriction endonuclease. Then fragment this with the restriction endonuclease produce fragments. Length polymorphism. Length means this is the length. Okay, this is a length. This length. Suppose this is A gene. In case of cow, the A gene are same. All the A genes are same. But in case of goat, the A genes are different. Okay, so we can easily identify the different organism. Okay, the difference between organisms by use DNA fingerprinting. This small fragment is called 2 to 10 kilobase pair is size, and this is called VNTR, variable number of tandem repeat. Okay, so in case of DNA fingerprinting, use VNTR, variable number of tandem repeat, or SNP, small nuclear polymorphism. Nuclear polymorphism. Polymorphism means in different organism this fragment is different. Okay, and different in number, also present different number. So here occurrence of restriction fragment polymorphism, phenotypic difference between individual, not by DNA fingerprinting we use the genotypic different, not phenotypic. Okay, availability of clone DNA. This clone DNA is not necessary for DNA fingerprinting. This clone DNA is produced by PCR technique. Knowledge of human karyotype. Human karyotype means arrangement of the human chromosome according to their size and shape. Okay, according to their size and shape. This process is called karyotyping. Next question. Assertion, replication and transcription occur in the nucleus, but translation occur in the cytoplasm. Definitely, in case of eukaryotes, replication and transcription process occur in nucleus. But in case of prokaryotes, there is no nucleus present. So transcription, replication, transcription, translation, all the process are occur in the cytoplasm. This is the right answer. Prokaryotes, mRNA is transferred from the nucleus into the cytoplasm where ribosomes and amino acids are available for protein synthesis. Okay, the ribosome and amino acid available in cytoplasm. This is also right answer. So, if both assertion and reason true, but reason is not the correct explanation of assertion. Both assertion and reason are true and reason is the, this is the correct answer. Why? After replication and transcription, for translation, the mRNA transfer into the cytoplasm, because in cytoplasm the translation machinery like ribosome, okay, and tRNA are available, and also amino acid are available. Okay, so A are right. satellite DNA is important because the satellite DNA means it is a repetitive sequence of DNA. So high degree of polymorphism. The satellite DNA in a single, suppose in a single organism, satellite DNA, the repetitive sequence are same. But in case of different organism, the satellite DNA are different. So through the satellite DNA, we can easily identify different organisms. So D is the correct. So high degree of polymorphism in population and also the same degree of polymorphism. Polymorphism means poly means many, morphism means morphology. And that means the different form of repetitive sequence so in different organism by satellite DNA. Where the satellite DNA is found? Satellite DNA present in the telomere portion of the, this is the chromosome. Clear? This chromosome in the telomere portion satellite DNA is present. In the centromere region satellite DNA is present. Clear? Next question, E. coli cell with a mustard gene, not mustard gene, this is a mutated gene of lac operon cannot grow in medium. This is not mustard, this is mutated. Lactose is a source of energy because the lac operon constitutively active the cell, not false. Because mutated, because lac Z gene represent beta galactosidase. So what is the function of beta galactosidase? It break down lactose into glucose and galactose. Okay. If the lac gene, if the mustard Z gene is mutated, so mutated Z gene means no functional beta galactosidase produce. Okay. So no breakdown of galactosidase. Okay. So they cannot synthesize functional beta galactosidase because they are mutated. In the presence of glucose, E. coli cell do not utilize lactose. Okay. And they cannot transport lactose from the medium into the cell. Okay. This is the function of transferase, beta galactosidase transferase, that is lac A gene. Next question 31. What would happen if in a gene encoding a polypeptide of 15 amino acid? So total number of amino acid is 15. Okay, 1 to 15 total number of amino acid. And 25th codon UAA is mutated to UAA. This is 25. So UAU mutated to UAA. UAA is the stop codon. The stop codon means here this is the active, all active amino acid. If the stop codon is present in 25th number chromosome, there is no protein no more protein are produced because protein translation is terminated in this region. So here polypeptide produce. Okay. What polypeptide? This is the active polypeptide. Active. The 24 amino acid, 24 two polypeptides of 24 and 25 amino acid will be pumped out. A polypeptide of 49 or a polypeptide of 24 amino acid will be formed. So a polypeptide of 24 amino acid are formed because after in this region or 25th position, due to the occurrence of stop codon, the polypeptide termination or translation termination occur. So no more polypeptide are formed. So this 24 amino codon are separate from mRNA. Okay, no protein are formed. So only 24 amino acid polypeptide are produced. The eukaryotic genome differ from the prokaryotic genome because the DNA is complexed with histone in prokaryote. No, in case of eukaryotes, only histones are present. In case of eukaryotes, in case of prokaryotes, there is non-histonic DNA are present. The DNA is circular and single-stranded in prokaryote. In prokaryotes, the DNA is circular and single-stranded. Okay. Repetitive sequence are present in eukaryotes. It is also right answer because there in case of heterochromatin region or telomere region or in case of in satellite region, the repetitive sequence are present in eukaryotes. Genes in the repetitive sequence is present very least in number but less number in prokaryotes. In prokaryotes also present but less number in prokaryotes. Genes in the former case are organized into operons. Genes of former case in both cases present, but in case of prokaryotes, circular and single stranded. Circular, it is the special character of prokaryotes. So this is the right answer. Next question: which is the most important factor for continuity of species from evolutionary point of view? In case of evolution means sudden and continuous changes of DNA sequence. Very slow but continuous process. Evolution is a very slow and continuous process. When evolution occurs, the DNA sequence or genetic sequence genotype changes, and at the result, for this result the phenotype also changes and new species are formed. So here only replication of genetic material is the appropriate answer for this evolutionary point of view, because formation of gametes only present in sexual reproduction. Okay, synthesis of protein, this is present in all living organisms. Proteins are synthesis normally, no problem, but replication occur in both asexual and sexual. Both reproduction, the replication are produced during the S phase of cell cycle. Okay. Next question is: select the incorrect statement. 6 codon do not code for any amino acid. Not 6 codon, only 3 codons: stop codon or nonsense codon or nonsense codon. These 3 codons are not code for amino acid. So 6 is the incorrect answer. Codon is read in mRNA in contiguous fashion. This is the right answer. In mRNA, codon are present in mRNA, that is contiguous. 3 codon function as stop codon. This is 3 codon function as stop codon. The initiation codon AUG code for methionine. The initiation codon is for methionine. So 2, 3 and 4 are right answer, and only 1 alone is incorrect answer. So here the D is the correct option. Next question: match the enzyme beta galactosidase. So what is the function of beta galactosidase? It's in the break. It is represented by LAC-Z gene. Okay, in operon that break down lactose into glucose and galactose. Okay, galactose. So beta galactosidase it increase the hydrolysis of lactose. Permease. Permease increase the permeability of beta galactosidase. Ligase, joining of DNA. And ribozyme. Ribozyme is a RNA. Ribozyme, one type of RNA that act as enzyme. Ribozyme is one type of RNA that act as enzyme, and it break the peptide bond formed between the amino acid. Okay. So here right answer A3, this is A3, B4 and C1, B is the correct option. Next question, which of the following events would occur in lac operon of E. coli when the growth medium has high concentration of growth medium with high concentration of lactose? The repressor protein attached to the promoter sequence and derepress the operator. The structural genes fail to produce polycistronic mRNA. Polycistronic means there are many cistrons or genes present, that is special character in prokaryotic cell. The inducer molecules binds to the repressor protein and RNA polymers bind to the promoter sequence. The repressor proteins binds to RNA polymerase, and so this is the suppose this is the lac operon model. Okay, here in the repressor protein, promoter region, operator region, this is Z, this is Y, and this is A. Clear? So lactose is a inducible operon. Lactose is a inducible operon. Inducible operon. So when lactose is absent, when lactose the repressor protein produced by transcription translation, repressor protein produced. If available amount of lactose is present, then lactose bind with repressor protein and repressor protein become inactive. So they do not bind to the operator region. So promoter region RNA polymerase easily move from 5' to 3' region, and here transcription occur, then protein synthesis translation occur. The repressor protein attached to the promoter sequence and depressor, no, the structural genes fail to produce false. The inducer molecule binds to repressor protein and RNA polymerase bind to the promoter sequence. C is the correct answer. Next question is, which of the following statement regarding HGP, Human Genome Project? Okay, the main purpose of Human Genome Project is to better known or better understand the gene sequence and the function and number of gene of human beings, through which we can detect different genetic disease and recover it through gene therapy, or by know the function of different gene, we can develop the medical science. Okay, this is the purpose of HGP. The HGP involved, so here which option is incorrect? Okay, so HGP is involved in the mapping and sequencing of model of organism E. coli mass muscularis. Okay, no, Human Genome Project, the main function is the mapping, chromosome mapping and sequencing, or to know the nucleotide sequence. HGP involves in finding the genes related to human disease in Arabidopsis very few number of genes are present. Okay, HGP involved in study of gene function of model organism, definitely right. HGP involved in greater understanding of human evolution by studying the gene in flies, roundworms and mice. Okay, because the technique used in Human Genome Project, okay, to know the mainly Human Genome Project is established due to know the gene sequence, number of gene and function of different gene in human body. Clear? So here the right answer is B. In DNA fingerprinting technique, probe is used for hybridization of DNA fragment. Suppose in case of, suppose this is DNA, through restriction endonuclease cut into small pieces, then these are separate on the basis of size, on the basis of size by gel electrophoresis, then it is transferred to nitrocellulose filter membrane, okay, in where suppose this is the nitrocellulose filter membrane, this is the DNA sequence, okay. This is the sequence of these DNA are unknown, but we can use cDNA. The sequence is known and also radio labeled, clear. This DNA probe is attached with this DNA, okay. Suppose in this DNA, if A is present, definitely it in opposite strand T is present. In this process, we know the DNA sequence of unknown DNA fragment, clear. So here DNA fragment, here radioactive DNA, single stranded radioactive DNA, that is called cDNA, complementary DNA. The sequence of complementary DNA is known and produced in laboratory from mRNA. DNA fingerprinting technique is based on structure of chromosome, identification of nuclear sequence present on DNA molecule, structure of nucleus. Now DNA fingerprinting means know the sequence, unknown sequence of DNA, identification nuclear sequence. Right answer. Gene regulation governing lactose operon of E. coli that involves the LAC-I gene product is the LAC-I gene means this is the involves the LAC-I gene product. LAC-I gene product means repressor protein, okay. Negative and repressible because repressor protein prevent transcription, okay. Positive and inducible because it can be induced by lactose protein. The negative and LAC-I means inhibitor protein that produce repressor protein. When the lactose is absent, this repressor protein binds with the operator region and do not transcription occur. So negative and repressible because repressor protein prevent transcription by binding with the operator region of the DNA. Next question, given below are the steps of protein synthesis. Arranging them in correct sequence and select the correct option. This is the steps of protein synthesis or translation process are haphazardly arranged here. So sequentially arrange. Okay, first charging of amino acyl tRNA. 3. Then attachment of large subunit, attachment of mRNA and smaller subunit of ribosome. 2. Then attachment the attachment of large subunit. 4. Large codon anticoding reaction is occur here. Then attachment of large subunit. 4. The linking of adjacent, okay. The correct option is D. So here first this amino acyl tRNA is charged, okay. Then next the small ribosomal subunit, attached to the start codon of the DNA. Next the charge initiator tRNA, that is methionine tRNA are attached to the initiation codon. Next the large subunit are attached to this small subunit, okay. Then form initiation complex, clear. Then this peptide bond, the amino acid are between the amino acid peptide bond is formed, then polypeptide chain is produced. Next, which of the following statement regarding human genome project is incorrect? The during human genome project, human genome, what is genome? Genome is total number of gene, total number of gene in haploid set of chromosome, haploid set of chromosome is called genome. Okay. The human genome consists 3 into 10 to the power 9 base pair and about 20,500 genes. Okay. Total number of base pair in human genome is nearly 6.4 into 10 to the power 9 base pair. In case of haploid set, 3 into 10 to the power 9. The average gene size is 3000 base pair and dystrophin is the largest known human gene. Yes, this is also right answer. Chromosome 1 contain maximum 2968 number of gene and Y chromosome has at least 231 gene. This is also right. Repetitive sequence are not. There are many repetitive sequence present in human genome. Near about 70% repetitive sequence in present. Repetitive sequence are present in human genome project by human genome. The incorrect option is D is the incorrect option. Next question, arranging the various steps of DNA fingerprinting technique. The first, isolate a DNA. Okay, first isolation of DNA, that is 4. Then the DNA is fragmented by restriction endonuclease, digestion of DNA by restriction endonuclease. Okay, small fragment. Then separation of DNA fragment by gel electrophoresis. Through gel electrophoresis, the DNA is separated on the basis of size. Clear? That is 1. Next, transfer the separated DNA fragment nitrocellulose filter membrane, then 6. Okay. Next, detection hybridization using label VNTR probe, variable number of tandem repeat. Okay, 3 number, and then detection of hybridization DNA fragment by autoradiography. The first 4, 2, 1, 6, 4, 2, 1, 6, 3 and 5. This is the correct option. This technique, first isolation of DNA, then extract the DNA, restriction endonuclease, fragmented, then gel electrophoresis, then transfer nitrocellulose filter membrane, then again radio labeled probe are hybridized with radio labeled probe, then auto X-ray with the help of X-ray autoradiograph. Next question, match the column 1 with column 2 and select the correct option from the given records. Alex Jeffrey 1983, maybe 1983, they discover DNA fingerprinting. Sanger annotated DNA sequencing process discovered. Jacob and Monet lack operon. Avery MacLeod transforming principle are. So A3, B2, Jacob and Monet C1. This is the B is the correct answer. Next question. Thank you. If you like this video, please subscribe and share my video with your friend. Thank you for watching this video.